Biotechnology is the manipulation of organisms or their components to produce useful products. It relies on tools such as nucleic acid hybridization, gel electrophoresis, PCR, and restriction enzymes. Together, these methods allow scientists to isolate DNA, study its sequence, and apply genetic knowledge to medicine, agriculture, and research. Genetic engineering introduces new genes into cells or whole organisms. In transgenic animals, the new gene is inserted into the fertilized egg, ensuring the trait is present throughout the body. In gene therapy, corrective genes are introduced into specific cells to treat diseases, replacing defective or inactive versions. DNA technology has revolutionized science and medicine. It enables forensic investigations, gene cloning for medical and industrial products, and the creation of genetically modified crops. DNA technology also uncovers human evolutionary history and has become indispensable in biological research. Biotechnology is not new. Humans have practiced it for millennia. Ancient societies used microbes to make wine and cheese and selectively bred plants and animals for desirable traits. Modern biotechnology builds upon this history with advanced molecular tools. Gene cloning produces multiple, identical copies of a gene. This involves combining DNA from different sources into recombinant DNA. Plasmids, small circular DNA molecules in bacteria, are often used as vectors to carry the target gene into host cells. The process of gene cloning begins with isolating plasmid DNA and the DNA containing the target gene. Restriction enzymes cut the plasmid in one place, and the target DNA is cut with the same enzyme. When combined, fragments align through complementary sticky ends, preparing for recombinant DNA formation. Recombinant DNA is created when DNA ligase joins the plasmid and target DNA fragments. The recombinant plasmid enters a bacterial cell, which then reproduces to form a clone. This clone contains many genetically identical cells, each carrying the target gene. E. coli is often used as a host in cloning experiments. Its plasmids serve as vectors to carry foreign DNA. Once transformed, E. coli cells reproduce quickly, generating large populations of bacteria carrying recombinant DNA. Restriction enzymes cut DNA at specific recognition sequences, often leaving sticky ends. These sticky ends enable fragments from different sources to pair with one another. DNA ligase then pastes the fragments together, forming stable recombinant DNA molecules. This figure demonstrates how a restriction enzyme cuts DNA at precise sites. The enzyme recognizes a short DNA sequence, producing fragments with sticky ends. When mixed with compatible fragments, these ends join to create recombinant DNA constructs. Genetically modified organisms, or GMOs, are created through DNA technology. These organisms are engineered to carry new genes, often providing traits such as resistance to pests or improved nutritional quality. GMOs are now widely used in agriculture, research, and medicine. Recombinant cells, particularly bacteria, are excellent producers of proteins. They grow quickly, are inexpensive to culture, and can be engineered to secrete proteins directly into their medium. This makes them valuable for producing medicines and industrial enzymes. Yeast cells, as eukaryotes, offer advantages over bacteria in biotechnology. They can process eukaryotic proteins more effectively and integrate foreign DNA into their genome. Yeast has long been used in bread and beer production and is now applied in genetic engineering to produce complex proteins. Mammalian cells are sometimes required to produce proteins with specific sugar chains. Examples include erythropoietin, which stimulates red blood cell production, and factor VIII, used to treat hemophilia. These cells ensure that human therapeutic proteins are properly folded and functional. This table summarizes examples of protein products produced through recombinant DNA technology. It includes therapeutic proteins like insulin, human growth hormone, and tissue plasminogen activator. Each demonstrates the medical value of biotechnology. Researchers are investigating the possibility of using entire plants or animals to mass-produce gene products. This approach, sometimes called farming, involves cloning genetically engineered animals or cultivating modified plants. While promising, it remains costly and technically challenging. DNA technology has transformed medicine. It has enabled the production of therapeutic hormones such as insulin for diabetes and growth hormone for dwarfism. It also aids in diagnosing genetic diseases, detecting infectious agents, and producing vaccines that protect against viral infections. Genetically modified organisms are reshaping agriculture. GM plants are engineered to resist pests, tolerate herbicides, or provide additional nutrients to combat malnutrition. GM animals are being developed for improved nutritional content and other beneficial traits. This slide highlights restriction enzymes and their central role in genetic engineering. These enzymes cut DNA at specific sites, allowing researchers to isolate or insert genes of interest. Restriction enzymes are essential tools for building recombinant DNA molecules. GM plants are designed to resist herbicides and pests, providing farmers with higher yields and reduced crop losses. Some are engineered to produce enhanced nutrients, such as rice varieties enriched with vitamin A. These advances aim to address global agricultural challenges and malnutrition. While GMOs offer many benefits, they also raise safety and environmental concerns. Risks include the spread of engineered genes to wild populations and the potential introduction of allergens into the food supply. Regulatory agencies carefully evaluate GMO safety, labeling, and impact before approval. Gene therapy holds promise for treating a wide range of diseases. By supplying functional versions of defective genes, this approach could correct genetic disorders at their root. Though still experimental, gene therapy represents one of the most exciting frontiers in biotechnology. Gene therapy is the alteration of an individual's genes to treat or prevent disease. It is particularly suited for disorders caused by a single defective gene. While challenges remain, successful therapies could transform the way we treat inherited conditions. This slide illustrates the process of gene delivery using retroviruses. A healthy human gene is inserted into the viral genome. When the virus infects patient cells, it inserts the corrective gene into the patient's DNA, allowing the cells to produce a functional protein. In 2000, the first human gene therapy trial targeted children with severe combined immune deficiency, or SID. 9 of the children showed remarkable improvement in immune function. However, some later developed leukemia, demonstrating both the promise and risks of this powerful technology. DNA profiling is the use of DNA analysis to identify individuals. By comparing DNA fragments from different sources, scientists can determine whether samples come from the same person. This powerful method has become essential in forensic science, paternity testing, and disaster victim identification. DNA profiling analyzes specific markers in non-coding regions of DNA. These markers vary between individuals, making them useful for identification. By amplifying and comparing these regions, investigators can establish genetic matches with high accuracy. The first step in DNA profiling is isolating DNA from the cells of a biological sample. Even tiny amounts of DNA can be recovered and processed. Once isolated, the DNA can be amplified and prepared for analysis. Polymerase chain reaction, or PCR, is a method of amplifying specific DNA sequences. It uses primers, heat, and DNA polymerase to copy a target sequence millions of times. PCR makes it possible to analyze DNA even from very small or degraded samples. This slide continues the discussion of PCR and its importance. PCR is fast, sensitive, and reliable, making it one of the most widely used techniques in biotechnology. Its ability to copy specific DNA segments revolutionized genetics, medicine, and forensic science. PCR, or polymerase chain reaction, is one of the most important tools in modern biotechnology. It enables scientists to amplify even tiny amounts of DNA into millions of copies. This process allows for rapid analysis of genetic material in medicine, research, and forensic investigations. Gel electrophoresis is a method used to separate DNA fragments by size. When an electric current is applied, DNA fragments move through a gel matrix. Smaller fragments migrate more quickly than larger ones, creating distinct bands that can be visualized and compared. This slide illustrates how DNA fragments of varying sizes appear after gel electrophoresis. Each band represents DNA molecules of the same length. By comparing patterns, scientists can identify differences between samples and analyze genetic variation. Short tandem repeats, or STRs, are repeating sequences of DNA that vary between individuals. By analyzing the number of repeats at multiple STR sites, forensic scientists can generate unique genetic profiles. This technique is highly accurate and widely used in criminal investigations. At a crime scene, DNA evidence can be collected from biological materials such as blood, hair, or skin cells. These samples are analyzed to identify suspects or confirm victims. DNA profiling has become a cornerstone of modern forensic science. This slide continues with the application of DNA profiling in crime cases. By comparing DNA from a suspect with DNA from a crime scene, investigators can determine matches with a high degree of certainty. This technology has revolutionized the criminal justice system. DNA profiling has provided crucial evidence in countless forensic cases. It is used to establish guilt or innocence, resolve paternity disputes, and identify disaster victims. The accuracy and reliability of DNA profiling make it one of the most powerful tools in modern science.